Epiboli. Epiboli describes one of the 5 major types of cell movements that occur in the gastrulation stage of embryonic development of some organisms. Epibolic movement is the way in which a layer epithelial cells spreads. This can be achieved in multiple ways. When undergoing epiboli, a monolayer of cells must undergo a physical change in shape in order to spread. Alternatively, multiple layers of cells can also undergo epiboli as the position of cells is changed or the cell layers undergo intercalation. While human embryos do not experience epiboli, this movement can be studied in sea urchins. Zebrafish. General movements. Epiboli in zebrafish is the first coordinated cell movement and begins once the embryo has completed the blastula stage. At this point, the zebrafish embryo contains 3 portions: an epithelial monolayer known as the enveloping layer EVL, a yolk syncytial layer YSL, which is a membrane-enclosed group of nuclei that lie on top of the yolk cell, and the deep cells DL of the blastoderm, which will eventually form the embryo's 3 germ layers ectoderm, mesoderm, and an. The IVL, ISL, and DL all undergo epiboli. Radial intercalation occurs in the DL. Interior cells of the blastoderm move towards the outer cells, thus intercalating with each other. The blastoderm begins to thin as it spreads toward the vegetal pole of the embryo until it has completely engulfed the yolk cell. The EVL also moves vegetally during epiboli, increasing its surface area as it spreads. Work in the ray finned fish Fundulus has shown that no large rearrangements occur in the EVL. Instead, the YSL also moves towards the vegetal pole, spreading along the surface of the yolk and migrating slightly ahead of the blastomers. Once epiboli is complete, the DEL, IVL, and YSL have engulfed the yolk cell, forming a closure known as the blastopore. Molecular mechanisms of epiboli. Cytoskeletal and cell adhesion components. Completion of epiboli requires the coordination of cytoskeletal changes across the embryo. The YSL appears to play a prominent role in this process. Studies on Fundulus demonstrated that the YSL is capable of undergoing epiboli even when the blastoderm has been removed. However, the blastoderm cannot undergo epiboli in the absence of the YSL. In zebrafish, there is a microtubule array in the yolk that extends from the animal to the vegetal pole of the embryo, and that contracts as epiboli progresses. Treating embryos with the microtubule, depolymerizing agent nacodazole completely blocks epiboli of the YSL and partially blocks epiboli of the blastoderm, while treating with the microtubule, stabilizing agent Taxol blocks epiboli of all cell layers. There is also evidence for the importance of actin-based structures in epiboli. Ring-like structures of filamentous actin have been observed at the leading edge of the enveloping layer, where it contacts the yolk cell. It is thought that a network of filamentous actin in the yolk might constrict in a myosin Roman II dependent manner. Treating embryos with the actin destabilizer cytochalasin B results in delayed or arrested epiboli. There is still debate on the extent to which the DL and EVL epibolic movements are active movements. The EVL contacts the YSL by means of tight junctions. It is thought that these contacts allow the YSL to tow the EVL towards the vegetal pole. Claudin E is a molecule found in tight junctions that appears to be expressed in the EVL and required for normal zebrafish epiboli, supporting this hypothesis. Additionally, zebrafish embryos that fail to make a fully differentiated EVL show defects in epibolic movements of the DL, IVL, and ISL, suggesting a requirement for a normal EVL for the epiboli of all 3 cell layers. The cell-cell adhesion molecule ecadherin has been shown to be required for the radial intercalation of the deep cells. Many other molecules involved in cell-cell contact are implicated in zebrafish epiboli, including G-alpha-12/13, which interacts with ecadherin and actin, as well as the cell adhesion molecule EpCAM in the EVL, which may modulate adhesion with the underlying deep cells. Signaling. The molecule fibrontin has been found to play a role in radial intercalation. Other signaling pathways that appear to function in Epiboli include the WNT/PCB pathway, PDGFP3K pathway, F-effrin signaling, JAK-STAT signaling, and the MAP kinase cascade. Other vertebrates. Epibolic movements have been conserved in vertebrates. Though most work on Epiboly has been done in fish, there is also a body of work concerning Epiboly in the African clawed frog. Comparisons of Epiboly in amniotes, teleosts and X. Leaves show that the key movement of Epiboly in the fish and frog is radial intercalation, while in amniotes it would appear to be cell division in the plane of the epithelium. All groups undergo cell shape changes such as the characteristic flattening of cells to increase surface area.